Good evening, class, and welcome to week 11 of semester. Only 2 weeks to go, so we're really on the home stretch here. And tonight we're going to look at the second half of lecture 15, genes, chromosomes, and DNA. So remember, we've already covered the first part of this lecture, and then after that, we'll get stuck into lecture 16. So let's move on to our discussion of DNA. So we've revised the structure of the DNA of the nucleotide. We've seen the differences between nucleotides found in DNA versus RNA, and we've looked at how they're situated and structured within a strand of DNA, where two strands bound together by hydrogen bonds forms the DNA component of a chromosome. And of course, that DNA is wrapped around histone proteins within the chromosome. Here we're going to step back a little bit in time to the 1940s, and let's think about the opinion at that time. Most people actually considered that DNA was too simple a molecule to actually be responsible for carrying genetic information that's transmitted from one organism to its offspring. And that's because DNA contains basically a sequence of only 4 different nucleotides, A, C, G, and T. Quite simple, chemically quite simple. Whereas proteins, on the other hand, consist of a sequence of 20 different amino acids. So protein is considered to be a lot more complex. So many people believed that it was proteins in the nucleus that carried genetic information that determines what that organism is going to be. But in 1944, Avery, MacLeod and McCarty published this famous experiment, and only the first of a series of experiments that demonstrated that it's actually the DNA component in the nucleus of a cell and not the proteins that actually carries hereditary information. They did this by observing 2 different forms of the same bacterial species. This is Streptococcus pneumoniae, and this is a bacterium that can cause pneumonia and bronchitis, meningitis, and various other infections. They used 2 different strains of the Streptococcus bacterium. One was virulent, that is, it caused disease, and it was called S for smooth strain. The other was not virulent, not disease causing, and it was called R for rough strain. So let's look at what they did with Streptococcus pneumoniae. They took the non-virulent bacteria that you can see here and injected those cells into mice. The mice remained healthy, no problem there. They injected the smooth, virulent strain into mice, and the mice died. Then they took the virulent cells and they heat killed them. So essentially, if you heat up bacteria to a certain temperature, you will kill the cells. The cells will basically break open, that is, they're said to lyse, and they can no longer express that virulence that causes disease. So when they injected the heat-killed cells, virulent cells, into mice, the mice remained healthy. Then, the classic part of the experiment, they took non-virulent Streptococcus cells and the heat-killed virulent cells, and they injected both of these together into mice, and the mice died. So how do you explain that? To understand the significance of that, we need to understand what bacteria can do. And I'll just grab my pencil. Right, here's an intact bacterial cell. When that cell dies, for example, as a result of heating it up to fairly high temperatures, the cell dies, it lyses. So it pretty much breaks open and all the cytoplasmic contents will leak out of the cell into the environment. And this includes its chromosome. So bacteria mostly have one circular chromosome that you can see here. Now, all the other cell contents will degrade very quickly, but DNA is a very robust, tough molecule. It's quite tough. And that means it can basically lie around and not degrade for a long time. And this is the basis of forensic science. They can pick up DNA from a crime scene that's been there for a long time. It'll still be intact, so they can basically characterise it and identify where it came from which individual it came from. All right, back to our DNA that's come from our virulent Streptococcus cells. Here we have our rough non-virulent cell. There we go. And what bacteria can do, they can make their membranes a little bit more permeable than usual, and they can actually take up naked DNA, so just DNA by itself, they can take it up from the environment. So they can take up this DNA from the virulent strain. Here it is. And now they have the DNA which will contain genes that enable the expression of virulence factors. That is a particular type of factor that enables that cell to cause disease. So what this has done basically, it's turned this normally non-virulent bacterial cell, and here is its normal circular chromosome. Now it's turned this particular non-virulent cell into a virulent strain of Streptococcus because it will express the virulence genes found on this DNA. So essentially this showed and a few further experiments that were done, this showed that it was actually the DNA component that carries information. Proteins would not enter the cell because the proteins will very rapidly degrade once this original cell lysis. Okay, let me clear that. Okay, a few years later, in 1953, finally the model of DNA was proposed by two scientists, James Watson and Francis Crick. And they published their proposed model of DNA structure in 1953. And this was a really big deal. There were many labs around the world that were basically racing to try to elucidate the structure of DNA. They knew about the different nucleotides, but they didn't know how it all fit together. And what happened, there's a bit of a story behind this, which I won't go into, but essentially everyone was using an experiment called crystallography. But they couldn't get it to work. They couldn't get it to work properly, and that's because they were using the DNA in a dry form. Another scientist called Rosalind Franklin actually carried out the crystallography experiment using wet DNA. So she got these beautiful results, but she didn't have time to analyse them. Watson and Crick apparently got their hands on her results. They analysed the results and they proposed the model of DNA structure. So quite a story. You can look that up if you like. What they proposed is that DNA is a double-stranded molecule twisted into a helix. They proposed that each strand comprises the sugar phosphate component of the backbone of the strand, and the attached bases are in the centre of the two strands of DNA. And those bases on each strand are connected to the other strand by non-covalent hydrogen bonding between the paired bases. Everyone already knew that the bases were adenine A, thymine T, cytosine C, and guanine G. And they also knew at the time the chemical structure of the nucleotide, that is the three parts, a phosphate group, a sugar, and a base. All right, so let's review our molecular structure of DNA. And I think this is a very nice image here. Clearly shows those two strands of DNA. And remember, those two strands together constitutes a molecule of DNA. So we can see 4 nucleotides on one strand of DNA, a cytosine, an adenine, a guanine, and a thymine. And of course, the sugar phosphate component of each nucleotide forms what's called the backbone of this strand. And the same thing applies to the other strand of DNA. We spoke last week about the 2 distinct ends of each strand of DNA. So we call this the 5' end because this free phosphate group here, which is not bound to anything, is attached to the 5' carbon of the sugar group. That's why we say this is the 5' end. And down here, we have the 3' end of this strand of DNA because we have a free, that is unbound, hydroxyl group that's attached to the 3' carbon of the sugar. We see the opposite on the other strand of DNA, 3' end up here and the 5' end down here. And that's because these nucleotides, as you can see, are positioned upside down relative to the opposite strand of DNA. We know how the 2 strands are bonded together as a result of hydrogen bonding. 3 hydrogen bonds always between a C and a G, and 2 hydrogen bonds between an A and a T. So because of this complementary base pairing, we could say if we knew how many cytosines were in one strand of DNA, then we would also know how many guanines were in the opposite strand of DNA. And the same thing applies to the other nucleotides. We've also seen that these bonds here that enable each nucleotide to be joined to the next nucleotide within a strand of DNA are called phosphodiester bonds or phosphodiester linkages. All right, bases include purines, A and G, and pyrimidines, C and T. We know that those terms arise because purines have a pair of fused rings and pyrimidines only have one. We've seen A and T connected by 2 hydrogen bonds, G and C by 3 hydrogen bonds, and the number of hydrogen bonds that are found between the nucleotides form the basis of that specific pairing. We've seen that bases are linked together by phosphodiester bonds, and that the nucleic acid strands of DNA have distinct ends. The 3' end of a strand of DNA has a free hydroxyl group on the 3' carbon of the sugar. The 5' end has a free phosphate group at the 5' carbon of the sugar. The 2 strands of DNA within a DNA helix are said to be antiparallel, which means the 5' end of one strand is directly opposed to the 3' end of the other strand. All right, and that brings us to the main part of this lecture, which is DNA replication. How does DNA build a copy of itself? But before we get into DNA replication, let's put that into some sort of context. Why does DNA replicate itself? Let's consider a single cell, and I'm going to draw a very large nucleus, and here we have a chromosome. That's 2 strands of DNA, and another chromosome, 2 strands of DNA again. Now, when this cell divides to give rise to new cells, this cell will essentially split to form 2 new cells, and each of these new cells has to have identical DNA to the original parent cell. Okay, I'll just draw some straight lines a little bit faster. Okay, so the DNA needs to be identical to what was in that parent cell there. After all, most of the cells in the body of an animal, whether it's a human or a fish, most of the cells in the body of a plant, regardless of the species, contain identical copies of these chromosomes. Identical. The DNA is identical. What's different between different cell types and different tissue types is how the genes found within those chromosomes, how those genes are actually expressed. That is, how they're switched on and off and at what level and so on. All right, so before a single cell divides into 2 new cells, which are called daughter cells, it has to replicate this DNA, so that this daughter cell receives the exact complement of DNA compared to the parent cell, and so does this one. All right, so now I want you to think about an example of cells that undergo cell division. So just based on what we've learned so far in living systems, can you think of any example? Okay, you might remember that we talked about a plant. Yep. There's the roots. Does anyone remember the cells here, cells within the plant that are constantly dividing?
Is it the meristem?
Meristems, correct, exactly right. So remember up here we have a meristem, shoot apical meristem, root apical meristem, and those meristematic cells are constantly dividing and giving rise to daughter cells. One which remains part of the meristem, the other daughter cell, which will differentiate to form a particular type of cell and tissue. Exactly. So when each of these meristematic cells divides, first it has to replicate its DNA so that the 2 new daughter cells that are formed each receive exactly the same complement of DNA. Absolutely right. So that's with plants. What about animals? Just from your own knowledge, can you think of a type of animal cell that's constantly dividing? Whether it's humans or fish, doesn't matter.
Is it stem cells?
Stem cells, absolutely. Everyone's familiar with stem cells. So we can think about stem cells that give rise to blood, the different type of cells that you would find in the blood circulatory system. We can think of stem cells that are constantly dividing to form cells of the immune system. Okay, so another example of cells, but of course animal cells. And skin. The skin cells are constantly being shed. Intestinal cells, stomach cells, the cells that you tested in one of your pracs. So you did a scraping of the cells inside your cheek. So those cells are constantly being shed and they're being replaced as a result of cell division. All right, so we can say that cell division is absolutely essential for all life, regardless of whether you're a plant or an animal. And even consider this very important single cell here. This is how we all start life, as a single-celled zygote, which arises from fusion of an egg cell with a sperm nucleus, and that produces a zygote. And that zygote will divide into 2 cells, but they stay joined together, and then they divide into 4 cells, and so on to form an embryo. Okay, and each round of division, of course, requires replication of DNA. Okay, so how does DNA build a copy of itself before the cell divides? DNA is said to be replicated semi-conservatively, meaning that each separate strand of DNA will provide the template for building the new strand. And this is possible because of the base pairing rules. Semi-conservative replication allows the synthesis of new strands of DNA with high fidelity. New DNA molecules consist of 1 old strand from the original molecule of DNA and 1 newly synthesized strand. So let's look at that process here, semi-conservative replication. So let's consider what's referred to here as the parental double-stranded DNA. This will be the parent cell, so this could be that zygote cell, for example, that's going to give rise to the embryo of an animal or the embryo of a plant. Exactly the same thing happens there. We start with a single cell. All right, so we can see our double-stranded DNA here, and you're familiar now with the base pairing rules and also what's meant by 5' and 3' ends of each strand of DNA. Okay, these will split apart, these 2 strands, and this strand on the left here it is here, will be used as a template to synthesize a complementary strand of DNA. So this DNA here will end up in one of the daughter cells after the parent cell divides, and you can see it consists of 1 old strand and 1 newly synthesized complementary strand. Okay, this strand here on the right from the parent will do exactly the same thing, and it will end up in the second of the 2 daughter cells with 1 old strand and a complementary new strand of DNA. Okay, but what you can see is that this molecule of DNA is identical to this one, and they're both identical to the original parental molecule. So this is called semi-conservative DNA synthesis. Okay, we're going to look at DNA replication in a lot more detail. And we're going to consider DNA replication in eukaryotes, so animals and plants. Okay.
Silvana?
Yes.
Are you going to tell us how they split apart?
Yes, absolutely, I will tell you. Yes, I will give you all the information now.
Okay, thanks.
That was just an overview. All right, so DNA replication in eukaryotes. Firstly, we need to unwind the DNA double helix because it's all coiled up, remember? So it has to be unwound, and this is possible through the action of an enzyme called a gyrase enzyme, and that will unwind the DNA. Then we need to split those 2 strands of DNA apart, because the enzyme that comes in and starts making the complementary strands of DNA can't work on that double-stranded DNA when they're joined together by hydrogen bonds. So another type of enzyme called a helicase will separate the 2 strands of DNA from each other, and it does so by breaking the hydrogen bonds. Okay, so that's those two enzymes. Now we're going to look at how the new strand synthesis takes place. And this takes place through the action of enzymes called DNA polymerases. Okay, now for this, I need to bring up my whiteboard. Here we go. All right, so we start off with our, whoop, that's not very nice, with our double-stranded DNA. Okay, and now that's been uncoiled and the two strands have been separated from each other. Well, they haven't been yet, but I'm going to show you how that happens. Okay, and I'm going to open it up. There we go. So I'm opening up just a portion of that DNA. So it's still wound together there. And I'm opening it right up here so I can show you what happens. Okay. Now, this top strand of DNA runs from 3' to 5'. Where are we? To 5'. And the other one is, of course, 5' to 3'. All right, let's put in some nucleotides. There we go. Some DNA nucleotides on this strand, and we're going to call this one the leading strand of DNA. And all this information is on that slide that we were just on. All right, so let's put in A, T, C, G, A, T, C, G, and so on. And you should be drawing this too, because there's nothing like drawing it to actually understand what's going on. Okay, now let's do the other strand, which we'll call the lagging strand. More nucleotides. So I hope you're all drawing this, and of course you're going to draw the complementary nucleotides on this lagging strand. So where there's an A here, there'll be a T here, there's a T there, there'll be an A here, whoops, and so on. And I'm sure yours will be much neater than mine. So a C and a T and an A and a G, C, T, A, and so on. Okay, I won't draw them all in. I'll just put another little line here.
Yvonne, what was the name of the top strand?
A leading strand. Leading strand, and the bottom one we'll call the lagging strand.
Thank you.
That's okay. All right, so we have unwound part of the double-stranded DNA. The hydrogen bonds have been broken. Now, before that enzyme called DNA polymerase, before it can start synthesizing complementary strands of DNA, that is replicate, that's what they call DNA replication, something needs to happen, because DNA polymerase needs what's called priming. It can't just chemically start adding new complementary nucleotides. So I'm just going to change the color here so it's a bit more obvious. So a different enzyme actually comes along, and the enzyme is called a primase enzyme, and it's going to add a short primer, a short RNA primer. There we go. This is a little piece of RNA which is complementary to the DNA. All right, so let's draw in our complementary nucleotides. Complementary to C in RNA is G. Complementary to G is C. What's complementary to A if we're talking about RNA?
Is it you?
So remember that U, correct. Okay, so remember there's no T in RNA. Instead, there's a uracil instead of the thymine. So we've got this little complementary RNA primer. It should be a bit longer than this, but I don't have much space. Now let's look at the ends of this RNA strand. If the DNA strand is 3' to 5', then that means this RNA, little piece of RNA, will be 5' to 3'. Okay, and that's important because you might remember that on the 3' end of a nucleic acid, there's a free hydroxyl group. And this is exactly what the DNA polymerase enzyme needs to be able to start synthesizing new DNA. Okay, now change my color once again. I might go for green. Now the DNA polymerase can come in, take advantage of the hydroxyl group and start adding nucleotides, DNA nucleotides that are complementary to the old strand of DNA. Here we go. There. And it can grab a T and put it in there because it's complementary to the A. So that means that there's a pool of nucleotides floating around that the polymerase enzyme can incorporate to synthesize this new strand of DNA. And this continues in this direction. And this is said to be continuous DNA synthesis because it only requires the addition of one RNA primer to set off the DNA polymerase enzyme. So this will continue in this direction towards this area here, which is called the replication fork. The replication fork. I'll just write fork because it takes too long for me to write. Replication fork, and as it approaches, it keeps replicating the DNA or synthesizing this complementary strand. As it approaches the replication fork, then this DNA continues to be unwound and separated from each other so that DNA synthesis can continue in a continuous fashion. So that's how the leading strand of DNA is said to be replicated. The other strand, the lagging strand, is a little bit different. Let's look at why. Okay, let's put a little RNA primer in here. I'll put it in here. Here's our little RNA primer. And now, because this strand of DNA runs from 5' to 3', then a complementary strand of nucleotides will be the opposite. We'll have the 3' here. Sorry, we'll have the 5' on this end, 5' here, and the 3' hydroxyl is on that end. So what do you think that means? So now the 3' hydroxyl that's required by the DNA polymerase is on this end rather than on the right side of the primer as it is up there. What do you think this means with regards to the direction of DNA synthesis?
The other way.
It goes the other way, correct. So the DNA will be synthesized in this direction rather than in this direction towards this replication fork. So what happens here in this section here? That still needs to be replicated. So what happens is that additional little RNA primers have to be inserted here and here, and so on. So then the DNA, or I should have changed my colour for the DNA. So then the DNA that's actually synthesised is synthesised here in these just little small fragments, and here, and so on. So the DNA synthesis on this strand here is said to occur discontinuously. And these little strands of DNA are called Okazaki fragments. So eventually what happens is that these little bits of RNA, these RNA primers in blue here, and this one up here, they have to be removed. And they have to be replaced with DNA nucleotides. So let's go back to our slide now and see if we've covered all the information that's required. All right, so polymerization or synthesis of the complementary strands of DNA requires a template to copy. Oh, have we got a question?
We can still see the whiteboard, we can't see the slide.
Oh, okay. Thank you. Okay, is that better?
Yep.
Oh, good. All right, so the new strand synthesis of DNA is performed by the enzyme DNA polymerase, but DNA polymerase requires priming in order for it to initiate the synthesis of that new strand of DNA. So to facilitate this, an enzyme called primase will come along and insert a small complementary RNA primer, normally about 5 to 10 nucleotides long, and this is inserted at a very specific sequence of DNA called the origin of replication. Basically, the enzyme will recognise a particular DNA site. So the short RNA primer is inserted and it has a 3' hydroxyl group on one end, as we just saw. DNA polymerase 1 is one of the polymerase enzymes. This is said to have proofreading ability. So it can actually check any added DNA nucleotides, and it's also responsible for moving those little RNA primers after DNA replication takes place. This particular polymerase enzyme, DNA polymerase 3, is the one that incorporates the new nucleotides in the synthesis of the new strand of DNA, and it does so only in the 5' to 3' direction on the strand of DNA that is being synthesized. So let's look at that once again. No, we won't look at it again because I've lost it. Fantastic. All right, so what we just looked at on the whiteboard is also illustrated here. But it's just showing one strand of DNA here, insertion of an RNA primer, which shows the free 3' hydroxyl group, and then DNA polymerase can start adding complementary nucleotides to synthesize a new strand of DNA. And then at the bottom here, we can see that the RNA primer has been removed. So DNA synthesis always proceeds from 5' to 3' on the strand that's being produced. So that leading strand that we saw at the top in our drawing, that strand is synthesized continuously. But the other strand, the lagging strand, the complementary strand to this one is synthesized in a discontinuous fashion. That is, the DNA polymerase enzyme has to work discontinuously in the direction away from that replication fork, and the DNA is synthesized as a series of small segments. So the primase enzyme has to attach a series of primers to that lagging strand of DNA rather than just one primer. Then DNA polymerase can extend the primers, but it does so away from that replication fork. The small segments of DNA that are synthesized are called Okazaki fragments, and these will be joined together later by another enzyme called DNA ligase. And this will occur once DNA polymerase replaces those little RNA primers with DNA nucleotides. Okay, here is DNA replication, and you can see why I tend to draw it rather than explain it as per this figure in the textbook. It's a lovely figure, but it's quite complex to try to understand. Someone's laughing. Why are you laughing?
Oh, I just see Mitch has written down, "This is fun."
This is fun. I didn't see him write that. Where did he write that? Are you having fun there, Mitch?
Yeah, I'm having heaps of fun. I actually enjoy this.
Oh, that's terrific. All right, so that's our DNA replication. Now, because eukaryotic cells contain a lot more DNA than prokaryotes, there's not just one origin of replication on each chromosome. There's actually lots of those open regions where replication is taking place. And this is in order for replication to take place in a timely fashion. Now, I only showed you one replication fork in that drawing, but there's actually two replication forks at each origin. And we can see this on the next slide. So this represents one eukaryotic chromosome, and we can see here 1, 2, 3 regions that are being replicated at the same time. Each of these regions is called a replicon, and only 3 replicons are pictured here. There's probably hundreds, if not thousands, of these on each chromosome. Each replicon that's being replicated has a replication fork here and another replication fork here. So that means replication is proceeding in both directions, not just one direction. Okay, we mentioned earlier on in this lecture that the two ends of a chromosome, there's one end here and another end here, these are referred to as telomeres. And telomeres are essentially regions that don't contain any genes. And there's a central region, or roughly a central region of each chromosome, that's called the centromere. And we'll come back to the centromere probably in second semester when we've all forgotten everything from first semester. And this is a region of the chromosome that's important in cell division, in the cell division process. All right, so multiple origins of replication, or replicons, in a eukaryotic chromosome. In a prokaryotic chromosome, such as a bacterial chromosome, normally you'll find only one origin of replication because there's much less DNA that has to be replicated. All right, and here's a summary of the important replication enzymes: primase, gyrase, helicase, DNA polymerase 3, DNA polymerase 1, and DNA ligase. So that completes lecture 15. And now we can move on to our third last lecture. in Living Systems. Lecture 16, the genetic code.
Silvana,
Yes.
this may be too much to go into, and I don't mind if it is. But where
Yes.
do the enzymes come from, or are they just floating around?
Very good question. Yes, they are being synthesized all the time. And in this lecture, we're going to look at how genes carry the information that enables cells to produce proteins, including enzymes.
Okay. So I
Okay?
should stop asking questions and just let you talk.
Yeah, no. Please ask the questions, because it might be something that I'm actually not going to cover, which might be useful to discuss. Yeah, so yeah, ask questions by all means. All right, so let's look at genes. So we know that genes are found within DNA, but what is a gene? How do we define a gene? Would anyone like to have a go at defining a gene? No?
No, not today.
No? Not today. All right, I'm drawing some double-stranded DNA here, and these little lines just represent the nucleotides. So I won't actually specify them. All right, so this is a little piece of a chromosome, and this region here, this particular sequence of nucleotides here, might be a gene. And there might be another gene up here and another gene down here. So you can see that genes are basically situated along each chromosome. Yep. And we can actually define a gene as being a specific sequence of DNA which encodes a functional product. So let's look at this gene here. This gene might be a protein coding gene. So it carries the information in the form of a code that will eventually produce a functional product, which is a protein. This gene here is a non-protein, might be a non-protein coding gene. It's going to, it might carry the information required to produce a particular type of RNA called rRNA, ribosomal RNA. And we'll come back to what that is. And here's another example of a non-protein coding gene. And it holds the instructions for a different type of RNA molecule called a transfer RNA. There we go. Okay, so these are all genes. They all encode or carry information required to produce a functional product, whether it's a protein, whether it's a type of RNA molecule. All right, so back in the day when people were first studying DNA and trying to understand the nature of hereditary information, through studies, I believe, on yeast, and sorry, I'll just clear this. Through studies on yeasts, they proposed a hypothesis. Someone proposed a hypothesis, and that hypothesis was that one gene provides the genetic instructions for one enzyme, and they called this the one gene, one enzyme hypothesis. One gene, one enzyme. And that's because they were studying enzymes. So they thought that all genes contained the instructions for synthesizing enzymes. This later turned out to be incorrect. and for a few different reasons. One, because not all proteins are enzymes. Enzymes represent just one particular class of proteins. For example, there are proteins that are structural proteins, and those proteins are also encoded by genes. Also, they found out that a complete functional protein might require 2 or more polypeptides to be joined together. So you might remember back to when we talked about the quaternary structure of a protein. There might be 4 polypeptides that combine to form a functional protein. So each of those polypeptides is encoded by its own gene. So you would need not one gene for that protein, but 4 different genes, each of which encodes its own polypeptide. All right, so this hypothesis was later amended to the 1 gene, 1 polypeptide hypothesis. And then eventually they proposed what's called the central dogma of gene expression. Central dogma of gene expression. Okay, and this is very simple, and it still will mostly holds true today. Central dogma of gene expression shows that there's a relationship between the DNA sequence of a gene and the amino acid sequence of the protein product. The central dogma says that information found in the DNA is used to form RNA, and this process is called transcription. Transcription. I'm getting worse and worse at spelling as I get older. Transcription. There it is. Okay, so the information found in the gene, in DNA, okay, is used to form a different type of code, slightly different code in a molecule of RNA, and then that information drives the formation of a protein. I should say polypeptide, but I'll just write protein here. And that part of the process is called translation. So the flow of information moves from DNA to RNA to protein. So, and this entire process of transcription and translation is referred to as gene expression. The information that's found within DNA and RNA that drives the synthesis of a specific polypeptide is deciphered using what's called the genetic code. And we'll come back to that later on. Now, even though we can see the flow of information moves in just one direction, it's now very well known that RNA can be reverse transcribed back to DNA. And this process can be done by certain viruses. So everyone's familiar with HIV, the AIDS virus. This is a virus that has a very unique enzyme that can do this reverse transcription process. Mitch, are you still with us?
Still here and loving it.
And love. He doesn't sound very convincing, does he?
I've convinced myself.
You convinced yourself. That's the most important thing. Good on you, Mitch. All right. let's look at transcription and translation. Over here in a eukaryotic cell, where do these processes take place? All right, here we have the nucleus of a cell. Here we have the DNA. Unfortunately, and I don't know why, it looks like it's a circular DNA molecule. This should be a linear DNA molecule, which is mostly found in eukaryotic cell nuclei. All right, a gene within this DNA is going to be expressed. So that means it's going to be used as a template to synthesize a molecule of RNA. So the code within the gene in the DNA will be transcribed into the code found in this RNA molecule. That RNA molecule has to be processed. And when it's finally been processed, you can see it's shorter now and it's had other modifications. It will be transported out of the nucleus of the cell, which means that transcription always takes place within the nucleus. It's transported out of the nucleus through the nuclear pores in the nuclear envelope into the cytosol of the cell. And then once it's in the cytosol of the cell, it is bound to one of our protein factories, the ribosome. The ribosome will actually decipher the code found within that RNA molecule, and as a result, it will synthesize a polypeptide. This process is called translation. So that's an overview of the gene expression process, which includes both transcription and translation. Now, exactly the same thing happens in prokaryotic cells, such as bacteria. But of course, because there's no nucleus, both processes occur together in the cytosol of the cell. Instead of here, transcription is compartmentalized in the nucleus, translation occurs in the cytosol. All right, a little bit of a review of RNA, but I think we can now move past this and start talking about transcription. So as we said, DNA acts as a template for the synthesis of a molecule of complementary RNA in a process called transcription. Only one strand of DNA will be used as the template, and only a single strand of RNA is actually synthesized. But just like DNA replication, transcription will proceed from 5' to 3' on the strand of RNA that's being produced. Nucleotides are added to this new strand of RNA according to our complementary base pairing rules. So let's look at transcription. Much more simple than the DNA replication that we looked at in the previous lecture. Very similar, but a lot simpler. Okay, so we have our 2 strands of DNA shown in blue. This region here has opened up. In other words, the hydrogen bonds have been broken and transcription has commenced. Here we have our new strand of new strand of RNA that's being synthesized in red, and this strand of RNA is complementary to this strand of DNA, which is referred to here as the template DNA strand. So have a look at the base pairing rules there. So remember, where there's a T in DNA, the complementary nucleotide in RNA will be an A, C in the DNA, G in the RNA, G in the DNA, C in the RNA. But here, this is the one that you need to be aware of because this is different in RNA. The uracil in RNA will be complementary to an adenine in the DNA. All right, so the RNA strand is being synthesized, and as it's being synthesized, it starts to detach from the DNA strand. Okay, note the DNA strand orientations also. So this template strand is 3' to 5'. So the RNA is the opposite, 5' to 3'. Okay, the enzyme involved in transcription, it's called RNA polymerase. Don't confuse this enzyme with the DNA polymerase that's used to replicate DNA. Here, RNA is being synthesized, so the enzyme is called RNA polymerase. RNA polymerase does not require priming, so there's no little primers that are inserted. RNA polymerase can do it all. It can separate the strands of DNA and it can start synthesizing this RNA molecule without any priming. So that's transcription. Very simple. Do you agree?
Completely.
Good. All right, let's recap. RNA polymerases do not require priming. The polymerase will bind to a specific sequence of DNA, which is called the promoter. I'll come back to that. Initiation of transcription then occurs by base pairing of the first RNA nucleotide. Elongation of the RNA continues by addition of complementary RNA nucleotides until a specific sequence in that DNA is reached. It's called a terminator sequence. And this is where the synthesis of the RNA stops and the RNA molecule is released from the DNA. Transcribed region is called a transcription unit, with the RNA molecule being called a primary transcript. All right, here we have the formation of the first phosphodiester bond, which initiates transcription. You really don't have to know any of this at all. This is just shown here for your interest. So it's just showing the DNA template strand, that one strand of DNA, and it's just showing an adenine nucleotide in the DNA and a cytosine, and it shows the first RNA nucleotide that comes in to bind to that adenine. And this first RNA nucleotide is a uracil. It's called UTP. The second RNA nucleotide that comes in is a guanine. It will bind to the DNA cytosine. This RNA nucleotide is called GTP, a GTP. Then the enzyme RNA polymerase enzyme actually catalyzes the formation of a phosphodiester bond between this phosphate from the GTP and the 3' hydroxyl group from the UTP. In catalyzing the formation of this bond, these two phosphates are released, and here it is, finished product, phosphodiester bond, and release of the two phosphates, which you'll recognise probably from one of your pracs, PPI. All right, once again, you can skip that slide completely. All right, transcription can generate 3 major types of RNA molecules. 3 major types. The one that we just looked at that's going to encode a protein is called, or will be called, messenger RNA, mRNA for short. This will carry the code that will determine the amino acid sequence of that final product, that final polypeptide product. There's another type of RNA that can be transcribed from a specific gene, and this is called ribosomal RNA, rRNA. We'll come back to that later. And here's our third major type of RNA, transfer RNA or tRNA. And I'll also come back to that later and describe that to you. All right, very briefly, let's look at transcription in prokaryotes, such as bacteria. In prokaryotes, transcription occurs in the cytoplasm, not in the nucleus. because they don't have a nucleus. A single RNA polymerase will transcribe all types of genes. Many of the transcripts, that is the RNA molecules that are synthesized, are said to be polycistronic. That is, one RNA molecule actually holds the code for more than one protein. In prokaryotes, both transcription and translation occur at the same time. They're said to be coincident or coupled. And that's because these two processes are not compartmentalized. So that's shown here, coincident or coupled transcription and translation in prokaryotes. So here transcription's taking place, that is the RNA molecule is being synthesized using one strand of DNA as the template, and before it's even been released, before it's even finished being transcribed, already ribosomes are binding to it. And the ribosomes will read the code found within that RNA molecule and as a result start synthesizing polypeptides, as we can see there. So that's coincident or coupled transcription. Okay, what about transcription in eukaryotes? Eukaryotic genes are usually monocistronic, which means that each RNA molecule that's transcribed will hold the code only for a single polypeptide. The eukaryotic nucleus contains a dark staining region, often referred to as an organelle, called the nucleolus. And this is the site where one type of RNA molecule is synthesized in great numbers. That is the ribosomal RNA. Again, I'll come back to that later. The three different major classes of RNA, that is messenger RNA, ribosomal RNA, and transfer RNA, are transcribed by different RNA polymerases in eukaryotes. All right, so transcription has occurred and a molecule of RNA has been produced, has been synthesized. But before this RNA can leave the nucleus and enter the cytosol of the cell, it has to be processed. There are three major types of RNA processing that occurs. First, both ends of that RNA molecule are modified. Here's the first modification: a methyl guanidine cap is added to the 5' end. All you have to remember is that a 5' cap is added. And I'll show you this in just a minute. It's actually just a modified nucleotide if you're interested. Second modification to the RNA molecule. A polyadenine or poly-A tail is added to the 3' end of the RNA molecule. And the third part of the RNA processing is called RNA splicing. And I'll show you that in just a minute. I'll come back to this slide. Here we go. Let's look at processing of RNA. And we're going to start at the top here. Here's our double-stranded DNA. But the nucleotides aren't indicated here. Apart from these three nucleotides here, ATG, which represent the very start of a gene. Here's the start of a gene here. And the last three nucleotides of the gene are shown here, TAA, and this is the end of the gene found within the DNA. So all these coloured boxes here, the orange and green boxes, these represent one specific gene. And you'll see that we've got introns and exons. I'll come back to those in just a minute. All right, for now, let's look at a couple of additional important regions. This region of DNA, which is to the left of the start of the gene, this is said to be upstream of the gene. And it's called the 5' UTR, 5' untranslated region. Upstream from that, this coloured box here, this is a region of DNA called the promoter. This is a specific sequence of DNA that's actually recognised by that RNA polymerase enzyme. The RNA polymerase always recognises the promoter sequence, it binds here and then moves along the DNA transcribing this gene. Okay, let's look at this region. At the end of the gene, this is said to be downstream of the gene. We have a sequence of DNA here that's called the 3' untranslated region. Then we have a region of DNA here, which is called the poly A site, and then a specific sequence of DNA here called the transcription termination site. So once again, RNA polymerase enzyme binds to the promoter, moves along the DNA, transcribing it to produce RNA, and once it reaches the termination site, it drops off. It stops transcribing and drops off. And the gene transcription has occurred. Here is the RNA that's been synthesized. Single strand of RNA, and before it's been processed, we refer to this as nuclear RNA or pre-messenger RNA. All right, that RNA, that nuclear RNA, has to be processed. It's processed by having a 5' cap added to it. Here we go, 5' cap. And at the other end, it needs to have a poly A tail added. So this A in brackets with a little n means that there's a number of adenines, about 200 adenines added to this end of the RNA molecule. That's our poly A tail. So all you have to remember, 5' cap on one end, 3' poly A tail on the other end. So that's two types of RNA processing. The third type is called RNA splicing. And now we go back to these green and orange coloured boxes. Let's go back up here to the DNA. Here's the gene, and there are some regions of DNA within a gene, these green regions here, that are not required to code for the protein. They don't form part of the actual coding part of the gene, so they need to be cut out, and they're called introns. Introns have to be cut out. The exons remain. They contain the code. All right, so let's look at how that's done down here. Here are the green introns. That's intron 1, and it's looped out from the RNA. Here's intron number 2. It's also looped out, and then they're cut away, leaving the orange regions, the exons behind, and these 3 exons are then joined together. Here they are, exon 1, 2, and 3 joined together. So now you can see that the final mature RNA, which is called messenger RNA, is much shorter than the original RNA molecule. Now, not all genes have those introns that have to be spliced out. Many do. They have different numbers of introns depending on the gene. There could be only one intron that has to be cut out. There could be many, many introns that have to be spliced out. All right, but what we're left with is the final messenger RNA, mRNA. And this can now be transported out of the nucleus and get ready for the next part of the gene expression process, and that's called translation. All right, does anyone need a break?
Yeah, wouldn't be a bad idea.
Yeah? All right, oh, it's nearly 8 o'clock. Yes, all right, shall we have 5-minute break?
Yep.
All right.
Silvana,
Yeah?
does this stuff ever stop blowing your mind?
No, it doesn't. It never stops blowing my mind, particularly what we're learning tonight.
phenomenal.
It is incredible. And as I was saying this morning to one of the students, it still amazes me that someone actually worked all this out.
Does anyone actually understand it, or is it just that we know what happens?
Oh no, this is really well understood. We can demonstrate this experimentally. Absolutely. This forms, what we're talking about tonight, forms the basis of recombinant DNA technology. Something that we'll talk about more in second semester.
Happens millions of times a day.
It's unbelievable. Yeah, constantly. This is happening all the time. If this stops happening, cells die.
Yep.
And the organism dies.
Phenomenal.
Yeah, this is ongoing. All right, I'll be back in 5.
Thanks.
Okay, are we ready to go?
Yep.
You ready? Is everyone back?
Yeah, never left, waiting for you.
Oh, you're waiting for me. You were so intrigued, Andrew.
Anthony, he's loving it.
I'm here.
Come on.
I am blown away, but,
Oh,
but
come on.
my
This is
goodness.
fantastic. This is great stuff. This is really always mind-boggling, no matter how much you learn about it. All right. Everyone back? Mitch, you still
Yep.
with us?
Yep.
Yes. Liz?
Yep, I'm back.
Oh, terrific. All right. So what we've looked at so far is transcription. That is, DNA was transcribed into RNA. So we keep talking about a code found within the DNA, and that code was transcribed into the code of RNA. Now we're going to look at the process of translation, where the code in RNA will be used to synthesize a specific protein. So what's the nature of that code? That code is called the genetic code. The messenger RNA will transfer information to a specific protein in the form of a code which is defined by a sequence of nucleotide bases in that messenger RNA molecule. Each amino acid that is going to form part of that new protein will be specified by 3 nucleotides in the messenger RNA, and each 3 nucleotides is called a codon. Let me show you something. Let me show you something here. So let's say that the nucleotide sequence in our messenger RNA, oops, it wouldn't be a T, it would be an A, went something like this. Okay, 5 prime, 3 prime. All right, let's say this was the nucleotide sequence in our messenger RNA molecule. These 3 nucleotides will be a codon. They represent 1 codon. The next 3 nucleotides in that sequence represents another codon, and so on and so on. And each specific codon holds the code for 1 specific amino acid. Since RNA is constructed from 4 types of nucleotides, A, U, G, and C, there are 64 possible codons. 3 of these codons, as we shall see, are called stop codons, and they specify termination of the polypeptide chain. Bear with me if that doesn't make sense yet. It hopefully will. Okay, I'll come back to this slide in just a minute. Here it is. This is the genetic code found within this table. On this side, we have the first nucleotide position in a codon. Let's say it was A, U, and G in the messenger RNA. So we look for the first nucleotide. Here it is, A. Then we go up here to the second nucleotide position, which is a U. There it is. So we use this as a set of coordinates. U, go down to where the A is. So we're looking at this box here. Then we go to the third nucleotide position, which is a G in the codon. Here's the third nucleotide position here. Where's the G? It's here. So once again, go across to that box, and there it is. There's our codon, AUG, And what does it specify? It specifies this amino acid, which is methionine. So whenever there's a codon in the messenger RNA molecule that reads AUG, this will encode the amino acid methionine, abbreviated to MET. Let's look at some other examples. Here's an amino acid called proline, and you can see that it's encoded by 4 different codons: CCU, CCC, CCA, CCG. So they all differ just in the 3rd nucleotide position, which is quite useful because if one of these is mutated, so where there's a U, if that's mutated to an A, it doesn't matter. It will still encode the same amino acid, still encodes proline. Here's another one, threonine, which is also encoded by 4 different codons. Some of them are only encoded by 2 different codons, as we can see here, for asparagine or for lysine. There's one here, tryptophan, which is only encoded by 1 single codon here. All right, here's 3 codons in yellow, UAA, UAG, and UGA. These encode, they don't encode amino acids. They represent what are called stop codons. And I'll come back to them a little bit later. And this is also an important one. AUG, this is referred to as a start codon, and it encodes the first amino acid that will be incorporated into the new polypeptide chain, and that amino acid is methionine. So that's basically how you read the genetic code. And I'll show you how this works in just a few minutes. But first, let's go back to the previous slide. Sorry, back to the slide before. All right, so we said that there are 64 possible codons. 3 of these codons, remember, are called stop codons, and they will specify the end of the polypeptide chain that's going to be synthesized. That leaves 61 codons to specify only 20 different amino acids. Therefore, most of the amino acids are represented by more than 1 codon. So we saw that some of those amino acids are represented by 2 or 4 different codons. Thus, the genetic code is said to be degenerate. We saw that particularly in the 3rd nucleotide position of a codon, a base change often doesn't change the amino acid that's specified. This is simply referred to as a synonymous change or a silent change. We can also refer to it as a silent mutation because it has no effect on the amino acid encoded. All right, translation. We're nearly getting onto this process. During the synthesis of a new protein, a ribosome will move along the messenger RNA molecule that it has bound, and it reads its sequence 1 codon at a time from the 5' end of the messenger RNA molecule to the 3' end. Each amino acid will be specified by the messenger RNA's codon, as we just saw, according to that genetic code, that table. Each codon found in the messenger RNA will pair with a sequence of 3 complementary nucleotides called an anticodon. And this anticodon is found on a different type of RNA molecule called a transfer RNA, or simply tRNA. Let's look at what the transfer RNAs look like. Here is one transfer RNA molecule. Always reminds me of a clover leaf. All right, here at this end, it will carry one specific amino acid, which will covalently bind to this hydroxyl group here. At the other end of this RNA, this transfer RNA molecule, there is a sequence of 3 nucleotides here, 3 RNA nucleotides, which is referred to as an anticodon. So transfer RNA molecules are carriers of amino acids. There's one type of transfer RNA molecule that carries one type of amino acid. That means there must be 20 different transfer RNA molecules, one for each type of amino acid. Let's look at the anticodon again down here. So this anticodon reads UAC. Let's now look at a codon that you would find in the messenger RNA molecule. Here it is, AUG. If you look up AUG in that table called the genetic code, you will find that this actually encodes the amino acid called methionine. And sure enough, what is this transfer RNA carrying? It's carrying the amino acid called methionine. Let's go back to the anticodon. UAC is complementary to AUG, which means that this particular transfer RNA molecule that's carrying methionine can actually bind to this codon within the messenger RNA. So this shows how this code in the messenger RNA can be translated into the code of proteins, the amino acids that make proteins. And we'll see that at play in just a minute. So each transfer RNA only binds the appropriate amino acid for the anticodon that it contains, and it can be recharged with another one of those amino acids after it's deposited its amino acid into the growing polypeptide chain. All right, before we get into translation, we need to consider the protein factories, the ribosomes. This is where translation takes place. Each ribosome consists of 2 protein subunits, a large and a small subunit. So here's a large ribosomal subunit, a small subunit. These 2 will come together to form one functional ribosome. What are ribosomes made up of? They're made up of several different types of proteins and at least a couple of different ribosomal RNAs. So remember we said that there was another type of RNA called a ribosomal RNA. They actually constitute the ribosome along with several different types of proteins. The ribosome provides a precise method of aligning the codons that you find in the messenger RNA and aligning the transfer RNA molecules that are bringing in the amino acids. And this ensures that the amino acids are synthesized in the correct order. Let's look at this ribosome here, and you'll see that in this region here, this is the region that will bind the messenger RNA strand. Here we have 2 binding sites, a P site and an A site. These are the 2 sites where the transfer RNA molecules will come in. That is those tRNAs that are carrying amino acids. All right, now we can start with translation. And there are 3 phases within translation. The first one is called initiation. Let's look at that on the next slide. Initiation of translation. So it starts here with the small ribosome subunit. It has bound our messenger RNA molecule. There it is, single strand of messenger RNA, and it's positioned it so that it will read this start codon first, AUG. AUG is complementary to this anticodon found in the transfer RNA molecule that carries the amino acid methionine. So only this transfer RNA molecule can actually bind here to the AUG start codon. Here we can see that the large ribosome subunit has bound to form a fully functional ribosome, and the transfer RNA carrying the methionine amino acid is situated in the P binding site. Okay, the next phase of translation is called elongation. And we'll look at that on the next slide. So just look at the left-hand side here, just this part of the diagram. And now we can see not just that first codon, the start codon, but the next codon in the messenger RNA molecule. It's triple U. So the only transfer RNA molecule that can bind to it is the one that has the corresponding complementary anticodon, which is triple A. If you look up triple U in that table called the genetic code, you'll find that triple U encodes the amino acid PHE, which is phenylalanine. So now we've got two amino acids that are going to start making up this new polypeptide, but they have to be joined together. And you might remember that amino acids are joined together by bonds that are called peptide bonds. Okay, so what catalyzes the formation of that peptide bond? It's actually, funnily enough, part of the ribosome. And this part of the ribosome acts as an enzyme. It's called peptidyl transferase. And it catalyzes the formation of a peptide bond between this methionine and phenylalanine amino acids. In doing so, the methionine will be transferred over to here so that it's bonded to the phenylalanine. That means this first transfer RNA molecule is now empty. It no longer contains an amino acid, so it's now free to leave. When it leaves to be recharged with another methionine amino acid, the actual ribosome will shift over to the right. Let's look at that here. Now we can see that it's moved over to the right so that the phenylalanine methionine containing transfer RNA is now positioned in the P site. Remember, over here it was positioned in the A site. Because the ribosome has moved over, now the next codon can be seen in the A site of the ribosome. That codon is CUG. So the next transfer RNA, which has the complementary anticodon GAC, has docked. CUG, according to the genetic code, encodes the amino acid leucine. Once again, peptide bond has to be formed in between here. That's done by the enzyme peptidyl transferase, which essentially will transfer these two amino acids over to the leucine. Let's just consider, before we go any further, the general structure of an amino acid, which you might remember. Central carbon, got a hydrogen, an R group. Just put the hydrogen up there. It's the variable R group. And we've got an amino group here and carboxyl group here. Okay, so that's one amino acid. Now we've got an incoming amino acid, and that peptide bond is always formed between, I can't write this fast enough, and here's the next amino acid. The peptide bond is always formed between the amino group of one amino acid and the carboxyl group of a second amino acid. Okay. So just keep that in mind. All right, so what's happened so far? Now we've got 3 amino acids forming our new polypeptide. So once these 2 have been transferred over here as a result of the formation of a peptide bond between them, this transfer RNA is now free to leave. Oh, and here it is. There we go. Oh no, that was the previous one. Sorry, we can see it over here in this diagram. There's the transfer RNA that's leaving, the AAA anticodon, and now the ribosome moves over again in this direction, so that this transfer RNA carrying the 3 amino acids is now in the P site, and so on. And this continues with the ribosome moving from in the 5' to 3' direction along the messenger RNA. This completes elongation, and now we get to the final phase of translation called a termination. And this happens when the ribosome moves to a codon called a stop codon or termination codon. So remember, in the table called the genetic code, there are 3 stop codons. They don't encode any amino acid, so there is no transfer RNA that can actually bind to this particular codon. Instead, the process, the translation process stalls here, and then other proteins come into play. These are called release factors, and they will enable the release of this newly synthesized polypeptide chain to be released from the transfer RNA. It will also enable the 2 ribosome subunits to be separated from each other. And here we go, large subunit separates from the small subunit. The messenger RNA molecule can be released, and it is very rapidly degraded after this. It's actually, in fact, it's been degraded the whole time that this is happening. But because it has that very long poly-A tail, the important part of the messenger RNA molecule is still intact until translation is complete. So RNA as a molecule is not robust the way that double-stranded DNA is. RNA is constantly being degraded, and new RNA molecules are constantly being transcribed. So there it is, formation of a polypeptide, which will go on to be folded, maybe combined with other polypeptides to form a functional protein. And now you can appreciate how the code from the gene in the DNA is transcribed into messenger RNA, and then that code is translated into the code of amino acids in a polypeptide. All right, here are the 3 phases of protein synthesis in translation. Once again, initiation, elongation, and termination. All right, but let's look at what happens to these proteins. Where do they go in the cell? And of course, how important are they? So if you're still not sure about the importance of proteins, consider this. Every possible process, every possible reaction, every metabolic pathway that you can possibly consider in a cell, in a tissue, in an organism is facilitated through the action of proteins. Every process. Proteins determine not only what occurs within a cell, but also what that particular group of cells, if you like, will become. Whether a single-celled zygote will divide to produce new cells that will form a human, or will it form a plant, or will it form a fish, for example. All of these processes are mediated by proteins. But let's look at where these proteins might be going within the cell. So let's start. So we've got a single cell. Here's the nucleus. Once again, we've got a linear piece of DNA. That's better. A gene within that DNA is transcribed into RNA. The RNA is processed through 5' capping, 3' poly A tail, and splicing out of introns to form a messenger RNA molecule. The messenger RNA molecule can leave the nucleus through a nuclear pore found in the nuclear envelope, and in the cytosol it is bound up by a free ribosome. Here translation occurs to form a polypeptide. Let's say that this is a complete functional protein. Where might it go? It might actually remain in the cytosol. There's lots of metabolic pathways that occur in the cytosol. One of these pathways is particularly important. It's called glycolysis, and it starts providing energy that's required to drive many, many reactions within a cell. So this could be one of the many enzymes that's required for that particular metabolic pathway. All right, it may, after it's been formed, this protein might be sent back into the nucleus. Why might that happen? It could be one of those histone proteins that the DNA wraps around. Or it could be a DNA polymerase enzyme that's required now to start replicating this DNA in preparation for this cell to divide. All right, where else might this protein go? It might be required in this organelle called the chloroplast. So within the chloroplast, we have these thylakoid membranes, and here are all the pigments and proteins that are required for the process of photosynthesis. So this protein might be one of those proteins that are embedded in these thylakoid membranes. All right, back to the protein. It might be one of those proteins that's embedded in one of the membranes of the chloroplast. Remember, every membrane has those integral and peripheral membrane proteins, some of which are important transport proteins, transmembrane proteins. Where else might it go? It may be sent to the mitochondrion. So again, it might be a protein that's embedded in the membrane. It might be one of the many proteins that's found in the mitochondrion itself. Lots of processes going on there also. All right, it might be targeted to any organelle within the cell. The peroxisome. Peroxisome contains lots of enzymes, so this could be an enzyme that's required in the peroxisome. All right, let's go back to our messenger RNA molecule. Rather than being translated by a free ribosome, that messenger RNA molecule may be bound by a ribosome that's attached to the rough endoplasmic reticulum. So when translation occurs, that polypeptide is transported directly into the lumen of the endoplasmic reticulum. Here it might be modified, then sent out via these transport vesicles and deposited into the Golgi apparatus. Further processing, packaging occurs, and then the protein is distributed once again in transport vesicles. Where does it go? It might be sent to the lysosome. So the protein might be one of those many hydrolytic enzymes that's found in the lysosome. Or the protein might be actually sent to the plasma membrane of the cell. Remember, the vesicle, that membrane enclosed vesicle will fuse with the plasma membrane and deposit the protein to the outside of the cell. So what might this protein be? It could be one of the proteins that's found in the extracellular matrix. And you might remember collagen, elastin, there's lots of others, and these need to be secreted by the cell to the outside. So here we have many examples of where proteins are targeted. And you can see that every process, every organelle, every part of the cell requires proteins in order to function. And this is only the tip of the iceberg, what we're actually seeing here. Okay, in summary, gene expression involves the flow of information from DNA to messenger RNA in a process called transcription. The messenger RNA molecule specifies the amino acid sequence of a polypeptide. And it does that because it contains codons. Remember, each codon is represented by 3 nucleotides. Polypeptide synthesis occurs at the ribosomes in the cytosol of the cell, and this process is called translation. The genetic code found within that table is a triplet code. That is, each group of 3 nucleotides will code for a specific amino acid. Or, as we saw, there are signals for termination, which we call stop codons. Proteins are targeted to all parts of the cell and transported by targeting mechanisms. And that concludes our lectures for tonight.
Okay.
How are you all feeling?
A little confused.
A little confused.
No, it's just going to require, I guess, a lot of rehashing and to commit to memory, I guess.
Yes, yeah. It's not easy to follow when you first learn about gene expression processes, and yeah, it's certainly worthwhile going over this again. This is gene expression is something that you do go on with in more detail in one of the second-year subjects called Applied Genomics.
Yep.
So it's certainly worth learning before you get to that particular subject.
Yep. Okay.
An amazing process.
It is amazing.
It's absolutely amazing. It really is.
A little factory.
Sorry, a little
Factory of its
Yeah.
own.
Absolutely, absolutely. And to think that this is just occurring all the time, constantly. Constantly, ribosomes are being made, genes are being expressed at different levels, in different cells, in different ways. There's so much more to this story, so much more to it.
So this is the meaning of life then, Silvana.
Absolutely.
No wonder I feel tired. All that
Yes. That's right. Imagine what the poor little cells are getting up to all the time.
Yeah,
that's.
They get a rest for those.
They never rest. Never rest. They're just always at work, constantly at work.
So Silvana, sorry,
Yes.
this might be a bit of a stretch, but if this always happens and our cells are always regenerating, why do we get old?
Why do we get old? Now that's something that we don't talk about in this lecture. But remember that I said that we have 2 ends. Well, this is what they think causes aging. Remember that I said that there are 2 ends to each chromosome called telomeres?
Yep.
Well, those telomeres, without going into too much detail, those telomeres seem to miss out. A little bit of the end of each telomere seems to miss out when it comes to DNA replication. So every time chromosomes are replicated, a little bit at the end of each telomere basically misses out on that replication process. So that means that the chromosomes get progressively shorter and shorter each time that they're replicated. And this is thought to cause the aging process.
Good.
So that's it in a nutshell.
Yep.
We'll have to try and fix that, eh?
Oh, we do, yeah. That's a good third-year research, student research project. Make sure those telomeres don't get shorter and shorter.
Get longer telomeres.
Yes, yeah. And that's why there's no genes in the telomeres,
Mhm.
because those telomeres always get shorter.
Okay.
So it wouldn't be any, wouldn't make sense to have genes there that will suddenly get cut off after a particular round of cell division.
Okay.
Hmm.
Interesting.
Yeah, very interesting. The meaning of life, absolutely.
Yeah.
Feeding ourselves. Huh?
That's right, yeah. All right, how's everyone going with their prac reports?
Oh.
I've got one done. I submit one.
Yes.
Yes, so
Oh, fantastic. Fantastic. Yeah, I saw a couple of early reports come in, which was great to see.
yeah.
Siobhan, I have a question, if I may.
Yes.
I'm struggling to find a way to open it. For instance, the acid-base one. Like,
Yeah.
what would be a good way to introduce the subject matter before we kind of start getting into the specifics of what we did?
Ah, so how would you start the introduction itself? Is that what you mean?
Yeah, exactly.
Simply tell me about acids and bases.
Okay.
Define them. Define them, tell me a little bit about them.
Thank you.
And remember, in-text references for
Mhm.
all of those bits of information that you will have in the introduction. They must all be accompanied by an in-text reference, and then every in-text reference that you use must correspond to a complete reference in the reference list. Okay, so remember, don't forget to get on to Ernie Easy Referencing, and I'll just type that here in case you haven't gone to this yet. Ernie Easy Referencing. There it is there. So that's on our library website. All you have to do is just put this into Google search. Just search for Ernie Melbourne Polytechnic. It should take you straight to the Ernie site. And this will show you how to reference correctly, both in-text referencing and the complete reference in the reference list. Okay, and if you've got any questions about that referencing process, just send me an email and I can help you out there. Okay.
Okay. Yep.
The people in the library are really helpful with that stuff as well.
Yes, yeah, that would be terrific. Yeah, absolutely. Okay, any other questions for tonight?
Nah, we're good.
All good? All right, well, have a good week, everyone.
Yep, you too.
So,
Thank
Siobhan, is this
you.
the last assessed shoot one we have to do this week?
Yes, it is. Oh, sorry. Yeah, I forgot about that. There it is. Yes, this is the last assessed shoot. There we go. Due, of course, next Monday, by next Monday at 6 p.m. Assessment weighting of 5%. Okay. So there you go. And then we'll have one final set of tute questions, which will be based on the last two lectures. So next week we will cover 1.5 of those 2 lectures, and then the remaining half of the lecture will be covered in the final week of semester. Okay, and then of course in the final week of semester we also talk about the final exam. Okay,
Thank you, Siobhan.
all right. Thank you, everyone.
Thanks.
Thank
Have a good
you.
week.
Yes, you too. See you next week.